conference and the invitation to speak at it. Can you see my main screen now with the title,
Yes.
Does Evolution Stop? Okay. So I chose that name because I thought it was a bit clever, and we'll see if it is or not. And I think everybody in this meeting will say, well, no, evolution does not stop. It continues always on. And the checkpoints I've seen in the immune system, they're kind of a stop and go point. So I'm going to cover a lot of this complex in the immunology in the middle. And at the end, hopefully it'll be a bit more fun with some videos of Tasmanian Devils, because everybody's been sitting through online seminars all week and basically for the last 18 months. So hang in there and we'll see them at the end. So I saw the pretty bird pictures and I felt woefully inadequate. So I threw in all these pictures quick from my previous life as a hyena immunologist. And that was a much different time in my life. I'm pretty much strictly a molecular lab immunologist now. So the outline, and I'll pop this back up so you can, intermittently throughout the talk, so you can follow along and see where I am. I'll first talk about the Tasmanian devil facial tumor disease, and I'll call them DFT1 or DFT2 because, as Chrissy said, there's two different transmissible cancers. Then I'll go into checkpoint protein interactions and immunotherapies, touching on humans a little bit, but mostly in devils. And then I'll talk about the checkpoint interactions and what actually happens when immune checkpoints interact. And then finally, I'll talk about a bit of vaccine development stuff. Hopefully that will be a bit more fun. All right, so this was the first known image of a Tasmanian devil with the facial tumor. It was taken in 1996 by Christo Bars, and nobody could have predicted at the time what would become of this. So we don't know if this was the index case. Very likely it wasn't, but it was the first time it was photographed. And it took about 5 to 10 years for people to really pin down that this was a transmissible cancer. So devils should have 14 chromosomes. These tumors have several chromosomal rearrangements, and they'd always find this in the tumor. They'd always find the same genetic mutations in the tumor for the most part. And they wouldn't match the host they were on. So the only logical conclusion is that these tumor cells are actually going from one devil to the next and to the next and to the next. So over many years, these tumors have infected thousands, maybe over 100,000 devils. And in 2014, Dr. Ruth Pai from our team at the Menzies Institute for Medical Research discovered a second type of transmissible cancer. So again, it's a facial tumor and it's on the face. So we thought it was going to be the same thing. And then when they did the genetics and karyotyping, it was a different tumor. So this one actually has a Y chromosome and the other one is from a female devil. So very clearly, another independent transmissible cancer. As Ninky said earlier, the first devil facial tumor was found in the northeast of Tasmania in about 1996 and basically covers the whole state now. The second one was found down in a peninsular region in southern Tasmania, and it hasn't spread very far yet. So we don't really know what is going to happen with that one, so it's a bit of a wild card. But they're pretty horrific tumors and still wreaking havoc on the devils. So this is a photo I took back in May, and you can see they're not just facial tumors, but there's large ulcerated tumors throughout the body here. This is DFT1. This one had nipples indicating very recently there had been joeys in the pouch, but certainly there's no room left in the pouch for joeys. You can see the different size of these two legs here. The one leg is just massively larger than the other. So I don't know if it's ascites fluid or tumors under there, but it was in bad shape. And this is a DFT2 cancer where the devil was still alive, but it was quite sick. It walked into somebody's backyard and they put a box over it and brought it into the vet. And you can see here, these are where the lymph nodes should be, and these are very likely metastatic tumors in the lymph nodes. So they're pretty destructive diseases. Two different recent papers, one led by Billy Lazenby from 2018, and then another more recent one by Callum Cunningham and Mena Jones. And they both show pretty similar things, that as the DFTD prevalence went up across the state, and that being this line here, that the population quite quickly went down. So the estimate was about 50 to 60,000 devils in 1996, and now it's probably somewhere between 10 and 20,000, with the predictions that the population is going to stay down unless we see a change. And there has been some good news that I'll touch on in a minute. But this is a heat map showing the number of devils per kilometer in 1996, with red and yellow being the high density areas. And you can see where the tumor first started, it quickly started turning blue. And then by 2019, most of the state is blue, indicating low devil density. And the forecast for 2030, based on historical data, doesn't look good. But there has been some very good news in the history of the devil facial tumor diseases. So Professor Greg Woods, back in about 2006, started studying the devil immune system. And by about 5 to 10 years later, was already trying vaccines. So based on a killed cell vaccine. So they vaccinated healthy devils and then challenged them with tumors. And unfortunately, the vaccine did not protect the devils from getting tumors, because you can see these lines going up indicating that the tumor was getting bigger. But what they tried then was using a live MHC positive tumor cell as an immunotherapy. And after the immunotherapy was given, there was quite striking tumor regressions. So within about 2 to 3 months, these tumors were gone. And they were able to do this in 3 different devils. So that was pretty encouraging. And there's a lot of things going on in that study, and those were lab strains. But then really good news has also come from the field. So the first natural tumor regressions were published in this paper, again by Ruth Pai in 2016. And they trapped devils, measured the tumors, and then 3 to 6 months later, or 12 months later, caught them in the field, and the tumors were smaller. And as Chrissy and possibly Ninki also showed, that this is a very striking example of a tumor regression. So this was picture A, and then picture B 3 months later, and there's just a big scar there. And the important message there is that the devil immune system is capable of killing these tumor cells, given the right stimulation and context. So that's what I'm really focusing on, is trying to stimulate the immune system to kill tumors. So we heard Chrissy's talk just a little bit ago, and one of the things that led to her studies on NLRC5 and C2TA was this recognition that when we looked at the transcriptome of genes upregulated by interferon gamma, PD-L1 was one of the most highly upregulated genes in the immune system. And for those of you that don't follow human clinical oncology very closely, the genes PD-1 and PD-L1 have really changed the way cancer is treated in the last decade. With a single monoclonal antibody targeting the PD-1 protein, making about $15 billion for Merck last year. So they're highly effective treatments in a subset of patients. So with that, I'll move into the checkpoint protein interactions in immunotherapy a little bit, and provide a bit of the background showing why we think these molecules are quite powerful in regulating anti-tumor responses. So the first immune checkpoint inhibitor was approved for use by the FDA in humans in 2011, and by 2018, Professor Jim Allison and Tasaku Hanjo were co-awarded the Nobel Prize for Physiology and Medicine. And I really love this, that they got this, because they were just 2 curious scientists, and they said, oh, we keep seeing this molecule on T cells, and we keep seeing this molecule on T cells. I wonder what it does. And they made antibodies that could block the interaction of that molecule or that protein with its receptors or ligands. And it turns out that that's a really powerful way of turning on an anti-immune response. So it's a nice study or a nice example of how basic biology studies can lead to really great biomedical treatments. This is an example of how powerful these treatments can be in humans. So this is a metastatic melanoma. It was treated with an anti-PD1 monoclonal antibody therapy. And after a few months, these pretty nasty tumors are basically gone. The tumors did develop acquired resistance eventually, and then she was further treated with radiotherapy and an anti-CTLA4 antibody, and the complete regression continued on for quite a while after that. So tiny little antibody that weighs about 150 kilodaltons can take care of this massive tumor. Is a pretty means it's a pretty powerful molecule. So my background before coming to Devils and even before studying hyenas was to study immune checkpoints in PD-1 and PD-L1. And what I'm going to show mostly today is flow cytometry data. I don't know that everybody does that, but it's a way of analyzing protein expression on a lot of cells in a very short time. So these machines can run about 20,000 cells per second through it and quantify the level of fluorescence coming off, and the fluorescence is linked to antibodies or proteins. So what I did for the Devils first was I cloned the Devil PD-L1 and PD-1 genes. I expressed them in Chinese hamster ovary cell lines. Just that's an easy line to make transgenic cell lines in, and they express xenoproteins quite well. So in this column here will be CHO cell lines that express the Devil PD-L1, and in this column will be CHO cell lines that express PD-1. Into all of these cells, I then added a recombinant PD-1 protein. So this is a secreted or soluble version of it, so it's not stuck to a cell in any way. So when I add it to the CHO PD-1 cell line, it shouldn't stick. It doesn't stick to itself, but it should stick to this PD-1 cell line. And that's what we can see here is in this, all the cells are down in the lower left quadrant, but in this one, it's shifted up, indicating that this PD-1 recombinant protein sticks to the PD-L1 on the surface of that CHO cell line. When I add in the anti-PD-L1 antibody, monoclonal antibody that I made, you can see that this shift is gone. So there's no cells, like up here we can see the cells in the top quadrant. In here there's no cells in the top quadrant, but we can see the population shifts over to the right. That indicates that this antibody is indeed binding to PD-L1 and it's blocking this receptor ligand interaction. If we do the reverse and we use an anti-PD1 antibody that I made, it binds to this soluble PD-1. It blocks it from binding to its target PD-L1 on the CHO cells, and it binds to the CHO PD-1 cell line that we were expecting it to bind to. So this is the basis of the Nobel Prize stuff in humans, and we've now got these in our hand for devils. So I don't know that a monoclonal antibody is going to save a lot of devils because you'd have to trap them and give them antibody injections every 2 weeks, but it's a nice tool to have in our toolbox. The other thing that we can do with these is we can see if the patterns of PD-L1 expression hold for devils like they do in humans. So what we did here is we used 3 different substrains of DFT1 and then 1 cell line of DFT2, and we treated them with interferon gamma that I made. And if we go across 3 days and then remove the interferon gamma, we can see in the gray histograms that the level of PD-L1 goes up with interferon gamma, and then we take the interferon gamma away and it goes back down. And this is the exact same pattern that we see in most human cell lines. So again, the data is converging, suggesting that devils look pretty similar to humans in regard to expression of these powerful immune checkpoints. Here's the only immunohistochemistry slide that I'll show. Some people like them. I like this one because it shows a really nice devil lymph node here. So in this, you can see the germinal centers quite clearly. And if your screen has a high enough resolution, you can see a bit of brown for PD-1 expression. It's always focused inside these germinal centers. So it's probably on T follicular helper cells, these CD4 T follicular helper cells with PD-1 expression. And we zoom in, we get pretty good membranous staining with the PD-1 antibody. For PD-L1, the expression should be pretty restricted to antigen presenting cells like dendritic cells and macrophages and B cells. So it's much more sparse in the lymph node. And if we zoom in again, we're getting good membrane and cytoplasmic staining. So they're pretty good antibodies. All right, so that was, about 3 years of work to make those antibodies and do those studies. And, we weren't that much closer to saving the devils. But what I really wanted to do is start studying all the immune checkpoints that were in clinical trials in humans. So this was kind of a half review, half analysis paper that I published in Frontiers in Immunology in 2017. And at the end of this paper, I was really sick of it, but it was the first time I felt like a real immunologist because I had my head around a lot of different genes, and I made each one of these proteins actually look like they're supposed to, with like 2 extracellular domains and things like that. But I didn't have a way to study all of these, and I was quite frustrated by that. And the other thing that was frustrating is that in humans, if you do a Web of Science search for cancer, you'll get 47,000 papers between, I think, 2006 and 2016. For immune checkpoint, you'll get 6,000 studies. In MHC, you'll get about 5,500. You'll get a bunch for mice and some for primates, but for almost every other species and clade across the tree of life, there's just no studies on cancer or checkpoints. And there's some on MHC, but just across the board, it's pretty low. There'd only been 3 studies on immune checkpoints in marsupials. I think at the time, those were probably all mine, and there's quite a few more now, and there's quite a few more on MHC. And again, most of these are on devils in the marsupials. So this frustration led me to try to come up with another way of making proteins to study the immune system. So the system that I came up with, I called fluorescent adaptable simple theranostic, or FAST proteins, and that might sound ridiculous, but using the acronym FAST has actually been quite common in our lab now, with at least 10 people in our group have made some of these proteins. And the nice thing about this method is that you genetically fuse your protein of interest to a fluorescent reporter. So if you go through the whole process of purifying a protein, at the end, you just take it over to your DNA gel box with a UV or a blue light, and you turn it on, and you can tell if your protein is correctly folded or not, because it will light up. So if something went wrong at any step of the process, it won't be yellow or red or whatever color you made it. So it's a nice quick validation to see if it's likely to work. So with this system, in a paper we published in Science Advances last year, we were able to confirm 7 receptor ligand interactions between 12 different proteins. And we confirmed that a lot of different colors could be secreted as soluble fluorescent adaptable theranostic proteins. With that, we've also used these proteins to make several antibodies. So we've made antibodies against 4-1BB and several others, some of them monoclonal and some of them polyclonal. So it's really opened up what we can do with the devil's immune system and try to manipulate these immune signaling pathways. So the papers are obviously published, and if you have trouble sleeping at night, I highly recommend reading the 49-page technical protocol. This will tell you exactly how to do everything. If you fall asleep before it's done, contact Chrissy or me or somebody from our team that's went through the process before. The other thing we found that we can do with this system is, instead of using a checkpoint protein in the FAST system, we can actually clone in the nanobodies. And for those of you who hasn't used a nanobody yet, it's way simpler than using an antibody. So an antibody, you need 2 heavy chains. So you need a variable region gene and a constant region gene, and then 2 different genes for the light chains. With a nanobody, you just need the variable region from the heavy chain, and you get a very good binding affinity. So what we do is we just fuse the DNA for a nanobody to the DNA for a fluorescent protein, and very quickly you have like an antibody, but it's a fluorescent nanobody that you can use directly from supernatant. So one figure in our Science Advances paper, and this work was done by Helena DeLuca, is we took a human PD-L1 sequence and we cloned it into the Chinese hamster ovary cell line again, and then we made a human antibody. So this protein sequence was published, and we just reverse translated it into DNA, and then made the nanobody fuse to M-citrine, which is basically a green fluorescent protein. And then we could take the supernatant from the cell line making the nanobody and add it to this target cell line, and we can see that it quite easily binds. And you just put the supernatant on, you do a quick wash, and you have your binding nanobody. And when we add that supernatant to a different CHO cell line that expresses human CD80, you can see there's no binding. So you can go from protein sequence online to having a fluorescent nanobody in about 3 weeks once you get the process down. And some of these nanobodies are published in the protein database online, and a lot of them are highly conserved targets they're hitting. So you might be able to find one for your species. Another thing we've been able to show with this system is that there's a lot of nanobodies published for SARS-CoV-2 and also for MERS that target the spike protein. So as part of a separate project, we had a regular CHO cell line, and then we had a CHO cell line that we transfected the spike protein into. And again, this spike protein was fused to M-cherry. So we can see that these cells shift to the right on these dot plots compared to the top ones, showing that the spike protein in M-cherry is there. And when we add the anti-SARS-CoV-2 M-citrine nanobody, it shifts up. So we were able to make these in about 3 weeks, just based on sequences that were published by other people online. So it's a pretty quick and easy to use system once you get your head around the cloning. Excuse me. One of the other things we were able to do with this system is that we had identified that the CD200 mRNA was very highly expressed in DFT1 and DFT2 cells. So we took the CD200 protein, fast protein that we made, and immunized mice and got polyclonal serum, and then tested 2 different DFT1 cell lines and 2 different DFT2 cell lines. And we could see that on all of these in the blue line, CD200 is highly expressed. And this is also an immune inhibitory checkpoint molecule. So it gives us another target for manipulating the immune system. But I think it will be more useful for what I'm going to show in this slide. So what we want to do is start thinking about how these devil facial tumor cells are metastasizing throughout the body. So to do that, they're probably going through the lymph system or the blood. So as a proof of concept, we spiked devil facial tumor cells into 100 microliters of devil blood. So there'd be about 1 million immune cells in there and about 10,000 tumor cells. And then we added our CD200 antibody to see if we could pull those tumor cells back out of the blood. We ran them through the flow cytometer, and we found that we could sort the DFT2 cells back out just based on the CD200, because the expression was above all of the immune cell expression. So we're not going to take this to the field yet, but we have 3 or 4 other markers we think we could use to co-stain, and we could start looking for metastatic tumor cells in devils in the near future. All right, so I think I'm at 19 minutes now. That should be running pretty close to time. Now I'm going to go into what will be the complex part of the talk. So hopefully you think that last part was okay. And the immune checkpoints are not as simple as it looks like here with one-to-one interactions, because when one goes up, another one goes down, and some of them like to bind in ways that you weren't expecting. So one of the things that our group has looked at a lot in the past couple of years is something that called intercellular protein transfer. And I got onto this first from a paper in Science by Omar Qureshi in 2011. And what they did is fuse CTLA4 to a red fluorescent protein. So wherever CTLA4 goes, the red goes. And then they had CD86, one of its ligands, fused to a green fluorescent protein. And when these two cells come into contact, these CD86 proteins start migrating around the cell in lipid rafts to the immunological synapse here. And if you look at them over time, there's more and more green over here, and eventually these little dots of green start showing up in the red cell. And what's actually happening is the CTLA4 protein goes up to the surface, it grabs the CD86, and then it brings it back down either into the endosomes or into lysosomes and usually degrades it. So that's what the transendocytosis is. And a more simple look at this is if you look at the cell that initially had the CD86, it's decreasing in green over time. And the CTLA4 cell is actually getting greener over time because it's capturing these proteins from the other cells. So I'll say this right up front that if you're looking at the mRNA expression in this cell line, there'll be no CD86 expression there of mRNA, but you're going to see a lot of the protein there. So it's essentially decoupling mRNA expression from protein expression. So we wanted to see if the same pattern held for devils. So what we did was we fused our checkpoint proteins to either green or blue proteins or orange or red in some cases. You do a bunch of different combinations of them in 96 well plates, and then you run them through the flow cytometer. And if there's no interaction, no intercellular protein transfer, they stay blue or they stay green. But if the blue cell is capturing the green proteins, it tips over this way. And if the green cell is capturing the blue, it tips over that way. And if they're exchanging proteins bidirectionally, they both start moving up into this upper right quadrant. So if we look at the control, so just a cell line that has a blue fluorescent protein and a green fluorescent protein, there's no interaction. There's not many cells up in here. But when we take CTLA4 fused to a green fluorescent protein and mix it with a cell that has its ligand CD80 fused to blue fluorescent protein, the blue is getting transferred over here. So the CTLA4 is capturing it and turning this cell blue, which is the same pattern we see in humans and mice. If we look at CD28, which also binds to CD80, this is a little bit different. It doesn't capture the other protein and bring it into the cell. It just leaves it on the surface. So it's called trogocytosis. And trogo means nibble, I think in Greek. So it's just nibbling away at the membrane, and we get a bit of bidirectional transfer there. This was published in Developmental and Comparative Immunology last year by Candida Wong. So as an evolution conference, I have to show some protein sequence alignments, I think, to keep everybody happy. Normally people roll their eyes at these, but these are CTLA4 protein alignments with human on the top, several other carnivores, and then I have Tasmanian devils on the bottom line. And I'll ask you to focus on two areas here. So in this dotted box, you can see this is 100% conserved. This is the receptor ligand binding domain for CTLA4. So it's right out on the tip of the protein, and this is how it binds to CD80 and 86. And this sequence is also 100% conserved in CD28, which binds to the same ligand, CD80 and 86. And what might be interesting to a lot of the other people here is that this is almost 100% conserved in birds also. A lot of reptiles and amphibians even have the same MYPPPY sequence. And then the other motif of interest, so this part's inside the cell, and the cytoplasmic domain is this YVKM domain. So it's a tyrosine, valine, lysine, methionine sequence. And this is responsible for cycling the cell to the surface and back and making the protein move around. So what you can do is you can do a genetic mutation study where instead of having the tyrosine either here or here, you simply put an alanine in there, which has very different biochemical properties, and it should completely change the function of the protein based on human and mouse studies anyways. So what Candida did was had the regular CTLA4 protein, and then she did one where she truncated part of the cytoplasmic domain, but left this key YVKM recycling motif. And then she made another one where she deleted that part. Then she made another CTLA4 protein where she substituted an alanine for the tyrosine in the receptor binding domain. This is what the normal CTLA4 CD80 interaction should look like, with CD80 being captured by CTLA4. The truncated one doesn't really make a difference, but when you delete or you change, when you delete this tyrosine part, it looks very different. So I think now what's happening is the CTLA4 is probably still capturing CD80, but it's just sitting on the surface of the cell and it's not taking it inside anymore. And when you change the single amino acid in the receptor binding domain, this interaction is completely gone. And that's that's a pretty powerful change. CTLA4 knockout mice, when they tried making them, they would always die as juvenile mice of autoimmune proliferative disorder. So this is a very powerful immune molecule, and that's why there's been such heavy selection pressure on it. There hasn't been any changes in about 200 million years in that gene. I won't go through all of this one, but I just want to highlight how well conserved these interactions are. So these are CTLA4 and CD8 and CD80 that we did for dogs. And you can see, I'll just focus on the CD28 interactions here, so that when you mix a CD28 cell line with CD80, you get this trogocytosis happening again with a bit of bidirectional transfer. But when you change again that single tyrosine to an alanine, it's gone. So we've done this in dogs, and we have a much more thorough study done in humans, just waiting on a former honors student to publish it, but now he's doing a PhD, so I don't think he's ever going to get around to publishing it. But we looked at all the different isoforms, and one of the take-home messages from this one is that if you look at mRNA expression, you really have to distinguish the different isoforms. So for dogs, there's 2 isoforms of CD80, but in humans, there's been, I think, 5 isoforms of CD80 and 7 of CD86, and they all act a little bit differently, depending what's in there. So you really have to dig down into the transcript. All right, so I think I have 4 minutes left. That should be about right. So I'm going to jump off of the immune checkpoints with the thought that all of that we've learned about the devil immune system will feed into our vaccine development program. So many of you are probably in Europe and much more familiar with this geography than I am, but in 1978, somebody came up with the idea to put a rabies vaccine in a chicken head and throw it down a foxhole. So the fox ate the chicken head and it got vaccinated against rabies. And it's become more sophisticated technology over the last 50 years, where you put the vaccine in a bait and you throw it out for the animal to eat. And that's what these are, 2 commercially available ones. But the red dots here show where rabies was in Europe in 1990 in terrestrial wildlife. This is Tasmania overlaid on it for scale. And the blue dots show where rabies is still in terrestrial wildlife. And it's probably not a perfect match. There might be things elsewhere, but the point is that this oral bait vaccine approach has worked really well for controlling rabies in wildlife in Europe. And there's a nice story on it in National Geographic from about a year and a half ago. The basic idea is you find something the animals like to eat, you make it very stinky so they sniff it out and find it, and then you put the vaccine or viral vector inside it so they get vaccinated. So we think this might work for the devil facial tumor disease, and we published an opinion paper on this in Expert Review of Vaccines last year. We think it might work for the facial tumor disease because, like rabies, it's an orally transmitted pathogen. It's transmitted during biting and during mating interactions and other social interactions, and it's quite often manifesting in the oral cavity first. So the tumor can get in through a gingival crevice or a wound, or possibly just hang around in the mouth until it finds a little crack to sit in. So if we were to get the vaccine in the mouth, it could penetrate through the same gingival crevice or wound, or even through the receptor on the oral epithelium. If we could do that and have the right tumor antigens coded by the viral vector, we could induce immune memory before this tumor ever arrives. So it would be waiting for it and hopefully clear it out as a preventative vaccine. But what's really great about this approach is that if the devil already has a tumor like this one, and you can see a little bit of ulceration there, I mean, it's a pathway for a potential vaccine to get right in there. You could have the virus directly infect the tumor and cause the tumor cells to die directly and make the tumor upregulate its own tumor antigens. So we think we could get a 1-2 punch out of this. And Dr. Amy Gilbert from the U.S. Department of Agriculture National Wildlife Research Center has been guiding us through this process. She runs a large rabies research project for the USDA, and she's fantastic to work with. So that's kind of the end of the immunology. And now I'm going to start talking about the bait development a little bit, because when I talked to the people that have made oral bait vaccines, they all said you need to start working on the distribution system immediately because it's going to be much harder than you initially imagined. So a company called Animal Control Technologies has supplied us with placebo baits, just to basically see if the devils like to eat them and which one they like the best and if it can attract them in. And they do like eating all these baits well enough in captivity, and we've seen a little bit of it in the wild, but we had some other species taking the baits from us. So we borrowed a automatic bait dispenser from the USDA. So what this has is a tube here, a magazine with about 20 baits in it. It pushes one out, and when that bait is removed, it'll kick another bait out about 40 minutes later. So we lured this devil in with some fish oil, We have the baits in there. It's actually a fish-based bait, and you can see it's like a little raccoon, and now it's licking up some fish oil on the dispenser. You got to really strap it down because they have very strong jaws. And if we give it a little time here, we think we will find it. So this was great because we had possums, wallabies, feral cats, pattymallons, rats, everything coming by this dispenser for about 10 days. And the first time a devil came by, if we're patient here, it gets its paw in there and you can see it pull the bait out and it'll land right here in just a couple seconds. So there's the bait. The times we've seen them getting the bait, they almost always do that. They pull it out and miss it. They don't use their eyes to find food very well. They're almost totally sniffing it out. But eventually it does pick up the bait, has it in its paws here, and it nibbles away. And that's another thing that we were happy to see is that they often hold the bait in their hands because if it's nibbling a vaccine, and hopefully the crumbs will spill into their hands and into their mouth rather than if they're eating it on the ground and all the vaccine gets mixed in with the mud. So that's been some pretty fun work where we're attempting to take our immunology into the field at some point. So take-home messages from here is that there's been good news from the field and from the lab that devils can have natural tumor regressions, and hopefully they'll develop some resistance and that will spread widely. We can now develop recombinant proteins fast to manipulate the devil immune system and works for other species. The checkpoint interactions are highly conserved across species, and that single amino acid changes can have a massive impact on overall immune function, and that mRNA might not always equal protein expression. I'm going to do a quick plug here for the Wild and Comparative Immunology Consortium. We're having a symposium at the Australia-New Zealand Society for Immunology on December 10th, I think, or 9th. So the abstracts deadline was today, but you might still be able to sneak one in if you're interested. And with that, lots and lots of sponsors to thank to make all of this research happen. So I'm happy to take questions and look forward to chatting people in the breakout room.